Good day to everyone joining us and welcome to today's Xtalks webinar. Today's talk is entitled Solutions for Recombinant Protein Expression. My name is Aisha Rashid and I'll be your Xtalks host for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. I'd also like to welcome our attendees viewing this presentation from our LinkedIn Live event, and I encourage you to submit questions as well via the comments tab there. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming to those who registered on xtalks.com. At this point, I'd like to thank ATUM, who developed the content for this presentation. ATUM has more than a 20-year history of innovation. The company provides services for biologics drug discovery and development, including gene sequence design and synthesis, protein and antibody engineering, and transient protein production through cell line development. ATUM uses advanced AI, machine learning, and proprietary algorithms to de-risk and accelerate time to market for biologics. And now it's a pleasure for me to introduce our speakers for today's event. Dr. Dominic Esposito has led the FN-LCR protein expression team for 2 decades, supporting the NCI, RAS initiative, and NIH investigators with access to recombinant proteins. With deep expertise in multiple host systems and combinatorial cloning, he has produced many thousands of proteins. Dr. Esposito holds a PhD in biochemistry from Johns Hopkins University and is an author on over 100 publications. Dr. Mark Welch has been with ATUM since 2006. At ATUM, Dr. Welch developed the Gene GPS machine learning technology for experiment-driven gene expression optimization. He currently leads gene, protein, vector, and strain engineering contracts for clients as well as internal new technology development. Prior to ATUM, he held positions at Applied Biosystems, Cosan Biosciences, and at Maxigen. Dr. Welch earned his PhD at the University of Colorado at Boulder for studies of the mechanism and evolution of fundamental steps in translation. Dr. Klaus Gustafsson is a pioneer of applying machine learning to build better biologics. He received his PhD in molecular biology chemistry from the University of Umeå, Sweden. And I do know I've probably pronounced that incorrectly, my apologies. And now, without further ado, I'd like to hand over the mic to our wonderful panelists today. You may begin when ready.
Good morning, everyone. This is really exciting, and we here at ATUM, we're super excited to have Dom Esposito here and talking about protein expression. And I think the really interesting part with this is how truly this changes technology in the sense that we have protein expression is fundamental to everything about in biotech, and it's been for forever the core that allows us to engineer a biological system. And how do you move protein X from one organism to another or maximize expression? And it's amazing that this industry, that's a $1 trillion industry, and it applies to everything from protein pharmaceuticals to agriculture to food industry to all these different things. And still, some of the fundamentals of how to maximize expression is still poorly understood. So we went back and looked at the origin of this, of course, and we all know the famous Genentech paper when they took human growth hormone and expressed it. So this is from 1977, and really the fundamentals of how to design genes hasn't changed that much since then. And it really most people approach this by looking at natural sequences, mimic them in one way or another, typically by picking the common codons, and then and tweak them to make it easier to synthesize. Typically means minimizing repeats. And we know, and we had lots of data now saying that that is by no means a guarantee for protein expression, and certainly DOM's been one of the key drivers for this over the last couple of decades. And but I think it's really important for the entire industry to go back and look at the fundamentals and how do we build all these things to make sure that we actually make decent expression. So we'll try to build this out as more of a discussion and a dialogue between really Dom and Mark, and more than a typical, someone droning over some couple of slides. So hopefully it will come across as more of a really interesting discussion about something that's fundamental to everything we do across biotech. So Mark, if you want to start talking about a little bit about what influences gene design.
Sure, and yeah, great to be here. Great to be here with Dom. I mean, I've known Dom, I think, for about 15 years now. We go back, known Klaus for 30 years. But yeah, I have the greatest respect for Dom. I mean, his experience in this field is almost unmatched, and his focus on education of the public is a wonderful thing. It really has advanced our field quite a bit, and I'm glad to be able to have this discussion today. And as Klaus said, we're hoping this is just basically a throw back and forth. So Dom, at any point in my slides, feel free to interrupt me, add things you think. I know a lot of what those are, and if I'm missing some things, you will catch me on them, I'm sure. So what I wanted to do with this first slide is basically just set up how we think about gene optimization. A lot of the talk today is going to be how to optimize genes, how to make genes express well, not necessarily always at the highest level, and this is something I'll hit on multiple times, and Dom will as well, I think. What is optimization? So when we're talking about heterologous expression or just use of synthetic genes, what's the goal? So I'm starting here on this slide, which is just the basic central dogma of biology. So DNA is transcribed to RNA, is translated into protein. For most of our customers, they're trying to make as much protein as they can. And all of what we're going to talk about today, it's about how do we make protein well being as much as possible, or as functional as possible, or works well within the context of a cell for modification of a pathway or modification of a cell line towards some goal, what have you. And most of the time, the genetic information is introduced at the DNA level, but you will know of many applications where that may be at the RNA level, for vaccines, for cases where you want temporary expression, temporary existence of that transformed cell. You instead of modification of the genome or the extrachromosomal genome, you might use RNA. And different constraints may apply, whether we're designing genes for something that will be expressed from DNA versus transcription. Now, as Klaus had mentioned, almost all current and initial and current applications of gene design are focused on the translation step. So, and that's what's shown here. There are a number of different factors that may affect translation. For the most part, people have focused on elongation. What codons might give you the fastest elongation through translation. Some papers will talk about context, how you juxtaposition certain codons may affect translation. There's plenty of talk about RNA structure, particularly as it might affect initiation, but also might affect translation through a gene. So the focus has been there, but it's a very small part of the entire picture. And what we found over the years, and what we try to do at Maxigen, is understand all the impacts of sequence on what you care about in the end, making more protein. So there are a number of things in the sequence that may affect transcription, initiation of transcription, elongation, the processing of your transcript. And there are a number of things that may affect the stability, both of the DNA in the cell, they click on one more here, and the degradation of the RNA, all these factors, and this is just an oversimplification still. The things that we're listing here in pink are, that are different elements of sequence that may affect these different aspects of gene expression. But there are a number of things, a lot of things we don't know yet, but plenty of things that we know that are important that are not traditionally incorporated into gene design.
So if you do that, well, how come you just don't take the natural gene and just put that in?
Yeah, and you know, so all this applies to whether you're expressing all natural genes in the cell, right? All are subject to these elements, and anything we're making heterologously, we're also subject to all these things. But we're asking, and this is another thing we'll talk about a lot, and Dom, I'm sure, has a lot to add in there. But what we're asking the cell to do is nothing close to natural generally. So both, you know, especially when, you know, the simplest thing that's different is we're generally asking for the cell to make a lot more protein than natural genes are usually expressed at. And these genes are not natural genes aren't designed for high expression, and hosts aren't designed to overexpress our protein of interest. So then the question is, for all these elements, how do you know how they impact your system versus in your application versus how they might impact natural genes? And that gets at the, how do you pull information out of natural genes? Can AI solve this by just looking at natural genes? Well, nothing in nature is selected for. So I think that based on these criteria and under these conditions. I don't know if you have anything to add there, Dom.
Yeah, so I mean, I do think that that's a really important point. A lot of people miss, I think, is that the idea that there are that organisms are naturally designed to manufacture all their proteins at high levels, right? And I think early on, one of the questions we got asked a lot is why do you bother doing gene optimization of human proteins if you're expressing them in human cells, or E. coli proteins if you're expressing them in E. coli cells? And I think the key is that what you just said, they're not meant to do that. And I think this experiment I'm showing here, which we did many years ago, I think really points this out. So the mammalian gene collection was designed to try and identify and clone every single protein coding sequence that existed in human cells. And in the process of doing that, they isolated these from cDNA libraries, but there were about 10% of the known human genes that they never could get out of cDNA libraries. So we took 6 of these sort of at random, and we synthesized them using the native human DNA sequences, and then we had them also optimized with these gene optimization protocols. And when they're expressed in HEC293 cells, which are human cells, it turns out that only one of the 6 native sequences made any protein at all. It's the one on the left in lane 3 that you really can't even see probably on most screens. And that was bio-Western, so there was very little protein. The other 5 showed no expression at all. But if you looked at the optimized sequences, all 6 of them produced protein that was visible by Western, and 2 of them showed enough protein that you could see on a Kamasi stain gel and made more than 1 milligram per liter of protein. And I think the argument here is there are a lot of native human sequences that were never designed to be overexpressed at 1 milligram per liter level. There are probably genes that are designed to be made at very low levels. So optimization is not just for, you know, proteins in a heterologous organism. It's basically to fix the things that nature's already designed to keep proteins from being overexpressed at large levels.
Yeah. So what are the issues that you're trying to engineer for? And just looking at the sequence, right? So what are the codons and what's the size and scope of this?
Yeah, I mean, as we mentioned on that first slide, there's a whole lot of things to consider in optimization. The other problem is there's a whole lot of possibilities in any normal gene. I mean, so everybody's familiar with the redundancy of the genetic code. And if you just got a regular size protein, say a 37 kilodalton protein, 330 amino acids, there's 10 to the 159 coding possibilities. That's all just variation at the 3rd position, but that adds up. And it should be pretty clear to people, we're not sampling 10 to 159 different sequences and scoring them all for optimization. So we have to be able to simplify the coding algorithm. If we're going to get anywhere near what's optimal, we have to have some understanding of what optimal means to be able to narrow that space, and we can't test all those. So we back up one more.
Sure.
I mean, just one thing to just keep in mind as well. I mean, not only do we have that many possibilities, it's all almost 3rd position. the 2/3 of the sequence is fixed just from the protein sequence itself. So in many cases, you don't have a lot of options for local sequence motifs. You want to remove a restriction site or whatever. So you need to know when you're doing this, how is that going to impact things like codon usage or other things that might affect expression. So you have restraints in specific changes you can make because of the protein sequence, but you have many, many different possibilities at the same time. So you really need to understand, how these different constraints, how these different requirements of the gene affect your goals, basically, the fitness of the gene in your application.
Dom?
Yeah, so I think Mark just mentioned this, that the codon usage is important, and it was a big thing in the beginning of sort of gene optimization, 2 decades ago. Everybody knows the organisms have different codon usages. There's a very big difference between bacteria and eukaryotes, especially with regard to codons that are GC rich. So I think people really in the initial days thought, oh, well, we'll just do gene optimization based on codon usage, and that's going to solve all the problems. But the problem is that there are a lot of different proteins, again, that are meant to be expressed at different levels. So if we just look at generic codon usage tables, that's based on the total complement of every protein in an organism. So again, not all proteins are meant to be designed and expressed at high levels. And I think this is a really fundamental bias in the idea of codon usage. And unfortunately, a lot of companies and a lot of people still do gene optimization simply based on the idea of pick the most common codons or the codons that are most commonly used in all the E. coli proteins. And that really misses a huge amount of that information that Mark showed on the first slide, which is really affecting the production of proteins well beyond just codon usage.
It really simplifies the algorithm, and that's
it does. It does.
Because now you just have one codon pick for each amino acid. There's only one optimal sequence, and then you can edit out some restriction sites and some other things that you don't want. But so yeah, you don't have to sample 10 and 159 space. But does that work? And it's one of the things that, our company, the reason we embarked on what I'll show you about our approach, how we look at this, is that we started making genes in our early days of the company. We've been around for 20 years, using, I think, and looking at costs, but basically what other people did. We didn't know, we didn't have data. We were experienced in engineering of the type that I'll talk about and that we apply now to codon usage optimization. But we just started making genes, choosing the best codons, hoping they work. And we got feedback pretty quick that it didn't work a lot of the time. And it's similar to what Dom was saying. When you try it out, when you use natural biases, it was hit or miss. So we thought we would do this in a more smart way. And we found, and we'll show you in some other slides, that we can't just pick the top codon. And we have to do this. We have to go in more naively, let the system tell us what works and what doesn't work. This one slide that Klaus pulled up here, it gives you an example too. I think it relates to what we're talking about with natural bias and what optimal means. What we did here is just we have 5 different codings of a gene. These all have the same UTRs. They're cloned into either a low copy or high copy vector. High copy, we're talking about hundreds. Low copy, we're in the tens. It's a big difference in copy there. We put them behind different RBS strengths. So copy number gives us different levels of DNA. RBS strengths give us different levels of translation from the RNA that's made. And then the third column here is induction strength. So we use promoters that have various different strengths that range over an order of magnitude down to just leakiness. And we looked at what the rank order of the genes were under these different pressures. And what you'll see, highlighted with the arrows there, those two rows are just change of copy number. But we see these patterns throughout any different type of pressure. But just changing the copy number, we change the rank of the genes. And
so there are a lot of different variables that involve initiation of transcription, initiation of translation, copy. Biology is complicated.
And just to give you a little background here, I didn't explain, but red is good here. So more red is higher expression. So what is optimal again? Well, optimal depends on the pressures we're putting on. And what we said before, these pressures, high copy number, strong RBS, strong induction. This is like, in this case, it's a T7 promoter in a strength in a ORI vector with a very strong consensus RBS. these are conditions that you're using to try to overexpress your protein. Those conditions look different if you just drop the copy of what optimal is. So what is the question of what optimal is depends on what the pressures are, depends on the application. And I'll tell you, it also depends on the protein that you're looking at too. And we can get into that a little bit later, because different proteins, different amino acid contents, different folding constraints, whether it's secreted or in the membrane, they all put different pressures on the system for what is optimal.
Is that something you see as well, Don?
Yeah, absolutely. I mean, there's even more to it, because it's frequently companies will show data which is simply expression of proteins, but the other factor to talk about here is the solubility and folding of proteins, right? So we don't always want to make a huge amount of protein very quickly, because some proteins can't handle the folding of that. So sometimes the highest rank order by expression is not actually what we want, and we have to dial back certain characteristics. So it's very much an experimental, empirically determined thing. It's which I think is why doing it this way, where you look at a variety of data and you can make a matrix of this, is so much better than just having one standard algorithm that just picks one set of characteristics, because what you need is very dependent on what the protein is you're working on.
Yeah, absolutely.
So Mark, how do you build such an algorithm?
Yeah, I mean, so this slide, it's a simplified version of how we actually do optimization. I told you, I mean, we have to consider many things at the same time. It's not just one score, not just codon usage alone that tells us what a good gene or a bad gene is. I mean, even if that codon usage didn't match host usage, if it was some other codon usage ranking based on whatever score, that's not going to do it. We have many different constraints on that's going to be good. And so we end up basically scoring genes in many different ways. We're going to have to score them relative to things that you care about for your application, the prevalence, say, of cryptic splice sites, things that might affect the RNA stability in various ways, things that affect the initiation of translation versus translational elongation. There could be other things, genetic stability elements, and how we avoid certain repeats, even for synthesis, might be a constraint, but how is that going to affect our ability to express these genes? There's a lot of things we have to score. The typical algorithm for us will have tens of scores. And that will depend on what the application is. So what I'm showing on the left is just an example of a few types of things we might see. Each bar is a different gene. This is a set of 20 I'm showing here. But in reality, when we're engineering genes here, we engineer populations. Again, I told you can't sample a 10 to 159. So what we do is we'll create thousands. We'll score them in many different ways. We have to weight those scores for that, depending on the application. So again, I said, we'll score them in codon usage and various parameters, but we weight those parameters based on what is important for the application. And then that tells us what are good genes and bad genes. And this first example just shows, maybe the best gene isn't the highest codon usage. I sorted these by codon usage 1 to 20, but when you consider all the scores, maybe you need something a little compromised in codon usage, but good in overall based on all the
codon usage
parameters.
And codon usage here is not necessarily the most common codons, right?
That's right. This would be a score of codon usage, which is determined experimentally. That's the layer that's missing here, but referred to with the pink here. How we weight these scores, the best thing to do is to, and really the only thing that works, this is what we found, is we have to experimentally interrogate. And that's what that previous slide was showing you. What's the best gene? Well, it depends on what your system is. Certainly depends on the host, but it depends also on how strong you're expressing. It depends on some constraints, whether you're delivering its DNA versus RNA will affect, because some scores only apply to DNA and some to RNA. And that actually, I'm making a little side here, but when it comes to codon usage, if codon usage were everything, the same rank you'd see for RNA versus DNA, right? And I'll tell you, I don't have data that directly shows this, but I'll tell you unequivocally, that's not true. When we use the same gene, same coding for RNA versus DNA, we get different reproducible rankings. And so we have to tweak our algorithms depending on whether it's going to be delivered as RNA or DNA.
So next slide here talks about, this is a nice example. You want to walk through that one?
Yeah, sure. So when I said we have to interrogate the host, we go in naively, and just say what works and what doesn't. And this example is a set that we did in HEK. We've done this for many different proteins now. We have several hundreds of genes that have been used to interrogate HEK and CHO for a number of different protein classes. But this just gives you an idea of the approach we're taking. So we go in, and I say naively, we go in as naively as we possibly can. We just say, here's a number of variables, and the variables in this set would be the rows here in this matrix I'm showing on the left. And we make a bunch of genes that sample those variables. And we vary the variables using gene synthesis. We can vary them in a very systematic way. So we try to, we don't want any covariation between the things we care about. And that can be multiple codon usages. It can be RNA structure. It can be anything that we listed on that very first slide. Vary them systematically across the set of genes, throw those genes into test. In this case, we did this initial work with GFP. We just looked at expression levels by FACS. And then correlate the data that we're seeing with the variables that we have, build machine learning models on that correlate to determine that correlation. And then that's our algorithm for expression. If we do this over and over again and on enough proteins, we have a robust algorithm that comes out of that.
And the cool thing with that is it's nothing out of just GenBank and run most common codons, but it's all empirically derived.
We're not making any assumptions about what works and doesn't. And that's why when we do that, we see some very unexpected things. And this is an example in E. coli, one of our first forays into this. We did this with a couple different genes, the same kind of process I showed you on the previous slide. But what we saw with those genes, when we looked at the correlation of the expression with typical measures of gene optimization, so CAI, what's shown on that x-axis here is the codon adaptation index. That's almost all algorithms that are used even today are seeking to maximize that index. The top one in the codon adaptation index means you're using the most frequently used codons in a subset of highly expressed genes in the host. So one would be the perfect gene. And you can see here there's basically no correlation at all. Our best genes have intermediate chi, but you can see that it's not really that you want intermediate chi. You can have bad genes and good genes at intermediate chi. And when you dig into the data, it's some amino acids want maybe more frequent codons, some want less. You don't know, and then there are other factors that beyond codon usage that are important. You don't know until you dig in. But if you build machine learning models, which we're showing on the right, you can explain the data. So it's not just random noise; it's predictable from the variables that we understand that are varying intentionally, and we can build models that explain that. So this, what's shown on the right, that's the basis for our algorithms that you would access today. So if you come to us, we're giving you gene designs that are based on experimental data, that based on what works under high load in that organism. Now we could build different types of models for you depending on what your application is, but that's our default algorithm. This is just one more example. I don't need to belabor this, but we've done this in multiple different hosts. This is an example in yeast for a human membrane protein. So in Saccharomyces, same kind of thing. We varied genes in a systematic way across a number of variants. We did this with Bob Stroud, a leading crystallography lab. So they want as much protein as they can get. It's hard to express membrane proteins. When you use standard bias, the arrows indicate a couple of versions of sort of typical algorithms at the time, whether we used normal yeast codons or whether we used a bias towards just the high frequency codons. Neither one was near optimal compared to what we saw when we varied the set systematically.
And I think it's very clear that it shows a huge delta between a poor expression and a good expression.
It's pretty clear that codon usage is important from that set. It's
yeah.
And,
yeah. So I think.
Yeah. Go ahead, Don.
This data, I think, is sort of speaks to that. All right. So we, years ago, we decided to look at a comparison of different algorithms for this in a number of different proteins. So these actually are 6 intracellular proteins, and this work is all done here in E. coli. And just to point out, these are the data shown here are actually purified proteins. So this takes into account expression and solubility and behavior and purification. So it's kind of a real world example. And so the native codons here are the ones shown in gray, and these are all human proteins in this case, made in E. coli. And then 3 different algorithms, the ATUM ones in green and 2 other companies that do optimization in yellow and red. And I think the take home here is that, you know, most of the time optimization does do something, but it's not consistent across different proteins, and it's definitely not consistent across different sources of optimization. And in some of the reason for that is what Mark just talked about. So a lot of companies focus very heavily on codon adaptation index. And in fact, when we used to optimize our own genes ourselves like 20 years ago, that's what we did because that's what the literature suggested. And in those cases, companies that basically just use the same codon for every single occurrence of a particular amino acid, we don't tend to see very dramatic differences. So like, for instance, the one in red here is often very CAI based. And so, you know, in some cases you see improvement, but actually in some cases like protein 1, the optimized gene actually is producing less protein than we see with the native sequence. And you can see that some proteins really dramatically benefit from optimization. Proteins 3 and 5, which make very little protein natively, get a really significant boost by optimization. So again, I think
and Dom, I think that's really cool because those are the ones where it really makes a difference, right? For 3 and 5, those are tough to do anything with unless you get good expression.
Yeah, I mean, protein 5 native was barely detectable. It was like 0.2 mg per liter. So yeah, it's a huge difference, and you know that makes a big difference if you're working on therapeutic proteins or if you're working with crystallographers who require a lot of protein.
Yeah. No, we love this data. Of course, this is very nice to get support for our algorithm that we developed. So it's cool to see. We really appreciate this one. Do you want to talk about the story?
Yeah, so I mean, I think this is another good example. In this case, we're talking about a human protein. And so again, a lot of people don't think you need to do anything with human proteins, but it turns out that cytokines actually are not highly expressed recombinantly, and they're probably not highly expressed in cells as well. And this, I think, is a really important example. So we were asked to make this IL-7 protein for some collaborators many years ago. And if you just focus on the Western at the bottom, it's a little easier to see because the protein's been glycosylated and kind of smeary in the Kamasi gel. But the native protein expresses in the supernatant at that left-hand amount under the S column at about 0.75 mg per liter. And so we had this sequence optimized. We actually had it done by 2 different people. We had it done by ATUM and we had it done by another company. And both of them improve over native, but the ATUM one was the most significant improvement. In this case, purified at about 12 mg per liter. So right there is a 16-fold improvement. And one of the things you see commonly with cytokines is that the native human sequences are very GC poor, which is unusual for human sequences. And I think it, at least in part, helps with the optimization here is that in both algorithms, these were optimized up to a much more reasonable GC content. But 16-fold improvement is obviously a huge deal, right? I mean, that's a massive difference in the amount of protein. And this particular client was interested in therapeutic production of these proteins. So that's you know, we're talking millions of dollars of difference in a yield improvement, 16-fold. And we actually went on the next slide, I think, to actually further optimize this, in this case, by doing something that I think Mark will probably touch on a little bit later, which is optimizing the promoters that we're using. Instead of just assuming that a CMV promoter is always going to give you the best expression, which is what most vectors out there are, we have a panel of promoters we tested. In this case, turned out that the EF1 alpha promoter actually gave better expression. And so ultimately, we were able to optimize this about 20-fold to get from 0.75 to 15 mg per liter of production by combining the gene optimization as well as the vector optimization that we're using.
Yeah, I think the best approach for a lot of this, especially if you have a protein of interest, a single protein of interest, you know, the one gene and then the one at a time, one factor at a time in engineering language approach is not the way to go. I mean, there's so many things, and I showed you on that earlier slide that, you know, what is a good gene depends on what load you're putting on, and it may also depend on some conditions. You know, one thing I didn't mention on the, well, maybe we'll get into my rant about why the single codon thing doesn't actually make sense when you think about translation in biology, but suffice it to say, all these things interact, and we'll see where we get on the discussion if we want to get into that. But so one factor at a time is not the way to go. I mean, if you wanted to do this with promoters, you might want to have variety, especially if you're invested in one particular protein. You want to have multiple promoters, multiple conditions of fermentation, and multiple genes. and see what wins out. Now, we think our algorithms are because they're focused on high production, and we've taken that slide I showed you where there's 5 genes, and we see that rank order under high pressure. That rank order holds into fermenters really well. So we did a collaboration with the same ones with Lonza. They put those into fermenters, got the same exact rank under high load when you put them into extreme protein production conditions. But I think the best way to do it is, as you're getting at here, is to try all these things, and one at a time is maybe not as good. If you really want to invest at the beginning in a particular protein, every 10% counts.
And also think that, I mean, any protein, like IL-7, I don't think a mammalian cell want to produce 50% of cell mass in IL-7. That's probably not a healthy existence, right? And you're wasting all this ATP on garbage, and every ATP counts. And if you're wasting ATP, you're, in an evolutionary perspective, you're dead, right?
Yeah, so I mean, our philosophy for many years now has been, you know, when people ask, when do you do this? When do you gene optimize, and when do you not do it? And like Mark said, do you start by doing one at a time experiments and then doing something else? Our honest answer is that if it's within the realm of affordability for the lab, I would do it all the time. And we, in fact, pretty much do that now. We don't do PCR cloning anymore. We do everything synthetically and have everything optimized. The biggest advantage for us has always been with bacterial work. There are just a huge number of problems in bacteria, both from codon usage, RNA stability, and a lot of other issues. And this, you can do things like use these rare tRNA plasmid containing strains like Rosetta and things like that. But frankly, nothing beats the level of improvement of expression that we get from doing full gene optimization. And for eukaryotic work, it's not nearly as dramatic as E. coli, where sometimes you just can't make a protein unless you optimize it. But generally, we still see improvements in both of those systems. You know, 1 to 3-fold, maybe sometimes more than that. And in some cases, like IL-7, if you have some weird bioinformatics approach, like the GC content's bad or there's repetitive sequences, again, it can be super useful in these organisms as well. Yeah, and I think it's important to also remember the other advantages to synthetic gene production, other than just codon optimization, is that you can play around a lot with the sequence. So you can put tags in. You don't basically, it eliminates the very labor-intensive process of designing primers, ordering primers, doing PCR, validating by sequence, because basically you can do all of this at one time. And you know, so we make a lot of chimeric constructs and sort of weird novel constructs that would be much more difficult to do by traditional methods. And you know, it's done for us. It's validated by the company. I don't have to worry about the QC. It's really straightforward. So we really like this process, and I think a lot of companies, especially these days, have realized this is much more cost efficient in the long run.
No, I think this is it's very clear that this is where things are moving. This is where we want to make sure we can actually, to go back to the whole discussion around biotech being $1 trillion industry, and it's to this day, a lot of people are still just cloning by PCR, and it just drives me bonkers. Or cloning by picking high common codons, and as you said, sometimes it works and sometimes it doesn't. It's a flip of the coin, and it's like you go back to your slide that you had, Dom, where you actually did a nice, clean, third-party 6-protein survey, and it just makes a huge difference. So with that, we'll conclude and then open up for questions from the audience. So let me pull out here. So the question, one question is, so to Mark, do you need to develop unique models for every single organism, or is it a Uber model that goes for similar types of organisms?
Absolutely, you need different ones for different organisms. So I said, you know, we don't know how many we've done now. We've done insect cells, so we have models for SF9, we've Saccharomyces, Pichia, Agaricus bisporus, a mushroom, AGK. So we looked at AGK and CHO. Largely, those are similar to each other. You know, coli, bacillus, and you get very distinct preferences when you interrogate those. we do see biases. If you use just one protein in the interrogation, you'll see a bias going to that protein, and it makes a lot of sense when you think about it. This gets toward my rant on the codon usage, the Chi argument. But what you see often is that the biases that are most important, at least with regard to codon usage, are biased by the codon or the amino acid content of the protein. You can imagine if you have a protein that's skewed significantly, say in serine, relative to average proteins in a cell, you're stressing the serine translational machinery of the cell more than you are for other amino acids, and you'll see that impact your codon bias. So you don't want to build these models just on a single protein that might be biased, or if you do, you want a protein that might be more average, but it's better to have multiple proteins that stress the extremes as much as possible. And if you have a particular protein class of interest, you definitely want models on that. So yes, different hosts absolutely have different models, and it's good to have models that are based on multiple different proteins that cover the base of.
And Dom, you said you worked with quite a few different expression hosts over at Frederick. Do you see any difference, or you mentioned that mammalians tend to not be as big a difference as you see in prokaryotes or E. coli?
Yeah, I mean I think that's generally true. I think the area that's been the most interesting for us has been insect cells. So we do a lot of expression in insect cells, and historically, insect cell codon optimizations were really poor, to the point where some companies, it was almost routinely lower expression when we used optimized sequence than when we used native sequence. And insects have a lot of weird codon usage, and they also have some RNA stability issues, particularly with baculoviral work in insect cells. So I do think one of the best things over the last few years is the development of much better algorithms for insect cells. So we actually use the ATUM SF9 optimization, and that works really well. And so I think, again, it proves the point that you need to have a machine learning based, iterative process. You can't just a priori kind of decide that, oh, this is the codon to use, this is that to use. That I think is what sets apart the companies that are really good at doing this from the ones that are just basically doing, hey, pick the best serine codon and go from there. So, but definitely organism specific is important. I mean, if we're really strapped for time and money, we might just go with a human optimization and use that in both systems. But I actually frequently order them in multiple different formats for the different host systems. They're very different.
Do you want to pull a slide down where we compared multiple hosts with proteins?
Let's do that.
Hey, Mark, I have a question for you. Do you guys see any difference if you compare algorithms for secreted proteins versus intracellular proteins?
So if you're looking at total, this gets to something you mentioned before. If you're just looking at total production of the protein, irrespective of whether it's secreted or soluble, then yeah, the algorithms are similar. You can have an algorithm. Most of our algorithms are based on just total production, right? But if you have to look at if you're looking at something that's restricted by, say, the secretion machinery, the algorithms, basically what I would say for those is optimal. There may be essentially a ceiling, or you may want to control expression at a different level. What's optimal in terms of protein production per gene may be the same, but what's optimal in terms of getting you functional protein may be that you need to throw in another constraint, like what you were talking about. You might tune the promoter down. Make sure you're using an optimal secretion leader, which is definitely a variable to play with, especially if it's heterologous organism. You want to play around with different secretion leaders. We've done quite a bit of that here. Definitely see different protein preferences for different leaders. And so, yeah, that's how I would approach it generally for something.
And you think that's because you clog up
membrane insertion or secretion, or because it's toxic, or because because you have a folding limitation. You overrun chaperonins or something like that. You may need to tune, but it's probably we haven't seen a case where you would tune that necessarily on the translational level. You use good output gene, and then you tune it either through RBSs or promoters.
want to talk?
Yeah, I mean, this slide, it just addresses one of the things that Dom was talking about in Klaus's question about, do we see different preferences for different hosts? I mean, so what I'm showing here is a set of genes that we made. These are just 48 green fluorescent dasher proteins genes. So they're all the same genes, but tested in 3 different hosts here, and each plot is just a comparison of 2 hosts together. So what you can see, I mean, is that there's certain genes that are definitely good in one host, not in the other host, and there are some genes that work fairly well in multiple hosts. And so I flagged a couple. The green one here is one that works well in all hosts. It's not the best, I don't think, in any host, but it works well. If you need a gene that you can pop between hosts, that one would work. And then there are some that only work in some hosts and are poor in another. And so navigating that, learning what the preferences are of the host can give you that wiggle room and tell you, these things are important, these things are not. If you try to just develop one on one host, you might bomb for another. But if you collect this information in multiple, you can navigate this space. If you need a gene, if Dom wanted something that he could potentially try in AGK and SF9 and coli and doesn't want to have to make multiple genes for each, we can give him that using the combined algorithm that looks at all those constraints for the 3 organisms. Save some money there, but it definitely shows you in slides that different hosts have different preferences. If you were just trying to go for the optimal gene, you'd come up with a different algorithm for each.
So I have a question here. Do we have any experience with the translational ramp concept described by Verma et al.? This is the whole discussion around that first, I think, 15 or so codons.
Yeah, and generally you have more rare codons in the beginning.
Yeah.
The idea is metering the ribosome occupancy of the cell, of the gene, so that you don't have collisions and so you're optimizing the use of basically your RNA space for number of ribosomes. I mean, that's correlated also, it just turns out, with, at least in E. coli, with use of rare codon, or rare codons are correlated with also less RNA structure generally in that region. So definitely natural genes tend to be biased that way. They tend to have more rare codons in the beginning, but it also tends to be lower structured as well, which helps in initiation. In models, it makes sense. In practice, I don't know that we're seeing correlation beyond the avoiding structure at the beginning.
I think one of the problems with a lot of these publications is that they're very heavily focused on GFP, and GFP is not really a normal protein in a lot of ways. And so we ourselves published some stuff before about GFP optimization, and I think it's dangerous to draw too many conclusions from small numbers of different proteins, which is again why I like the algorithmic approach, because there's just a lot of things which are very selective, and GFP in terms of fluorescence is a very different thing than a lot of other proteins. So,
and I think what we do see is that you don't want to. I mean, the first 15 or so codons is more an on-off switch than an actually a driver of titration. Just looking at the underlying data what we have over the years.
We still see a lot of variation in genes when we already taken, if we normalize that initial segment, avoiding structure and have fixed sequence, there's still a lot of impact codon choice beyond that. In fact, I might even have a slide there, we don't necessarily have to go to it, but when we've analyzed genes that vary, that are still normalized at that front end, we see that if we just swap gene segments, so we'll make hybrids of genes, crossing segments in the middle and the end of the gene, and we see that largely a number of these elements that are important are additive. So we'll see contribution from things at the tail end, the middle, and the beginning of the gene. So it's not if you analyze everything in aggregate, you're going to see the things that are going to stand out are out of the noise are going to be those 5' effects often. But when you look at it in the individual genes, you'll see all of the things that I talked about, elements that are important for expression. You see those distributed throughout. You see important impacts of those throughout. So that's the 2 types of data that are out there. For people are focusing on one gene, you know, like if it's all GFP, or people are focusing on many different sequences, many natural sequences, both have their drawbacks to some extent, because you lose some information in the noise of looking at many different proteins, many different codings, but you also, you know, lose information if you're just looking at one protein that may not apply when you're looking at it different. So that's why we try to do both here.
So here's a great question. This is my favorite question by far. It will be, no matter what other question we get. But another question, for applications like gene therapy, are there expression variations based on cell type or tissue type?
Someone asked that question.
Yes.
Funny you should ask. Do you want to go? I have a slide on this. So we did a little bit of work at the Wilson lab at UPenn on this. So when we heard
this one,
yeah. So again, we had a number of variants of a gene. This is a particular this protein is a particular protein of interest for gene therapy that their lab is studying. We made a number of variants based on our algorithms that we developed from the studies on different proteins, like the first slide I showed you when we studied HEK preferences. So we made some variants that we expected to be different from each other based on those studies on other proteins. And they took those and actually introduced them into mice and into different tissues in mice. And on this slide, we're comparing what they saw in liver, muscle, and bronchial tissue compared to what you see in HEK. And what you can see in this data is there's very different preferences depending on the tissue you go into from what they saw. Particularly, we see something very different with you know, variants 2 and 3 looking strong in liver and poor in muscle, but the other, you see it the other way around, variants 7 and 8 are quite good in muscle and different in liver. And there's a lot of evidence if you fish through the literature that, yeah, even just for tRNA differences, the balance of tRNAs, synonymous tRNAs, and the differentiated tissue can be very different. Certainly miRNAs, a lot of other regulatory differences cell to cell. How tRNAs are modified, that gets at Klaus's heart, because Klaus is an old tRNA mod person. There's lots of differences in regulation that are directly related to the translation machinery and codon preferences. And that could be what we're reading out when we do this. It's hard to do these large scale studies that I've talked about if we're going to do them in mice and in different tissues. So having good representative cell lines would be great to really flesh this out. Wow.
And I think that to go back to the question, gene therapy, of course, is going to be mission critical to make sure not only that it's one thing if you do a replacement of the exact gene, but if you're trying to engineer now different modalities and different functions, that's going to have profound effects.
There may be cases where you're not trying to express the protein as high as possible, and you just need to replace it in a natural level, and so there might be different constraints on the gene for those applications.
So, so here's a question. We've clearly seen advancement in expression of difficult to express proteins over the last 10 to 15 years. Absolutely, I would think almost 30 years now. So where do you think we'll be in the next 10 or so years? Dom, you want to take that?
Yes. Well, I mean, the problem is that the targets are becoming more challenging too now, right? So I think we've killed all the low-hanging fruit. So, you know, from my point of view, a lot of what we work on are signaling complexes now. So we're not talking about expressing just one protein. We're talking about expressing complexes of proteins. So that brings up a whole nother level of difficulty in making sure that we're expressing all the proteins at a similar level and can, you know, actually purify out these complexes. But again, I think we're driven a lot now by the ability to do optimization, to control those levels, and like Mark said, to tweak promoter strengths and things like that to try and balance the level of expression. So, I mean, I think things are getting better as far as that goes. We're seeing more and more data from these large protein complexes. A lot of the downstream technologies require a little bit less protein now. Things like cryo-EM have really helped to reduce the required levels of protein. So I think that's a positive as well. But you know, proteins are going to be challenging. They're all very individual. They all require a lot of sort of customized attention. So I think we'll all still be in business in 10 years. That's for sure.
Yeah, I think we're still just scratching the surface, I think, in terms of understanding these variables as well. So we can do this. Our experience is the way we're approaching this is the way it just have to go. And what I mentioned before, I mean, I don't think AI can. There's nothing for AI to work on right now to be able to design the gene for you. You can't just copy natural genes, and that's all that we have out there, natural genes and their natural expression levels. So you need to be feeding AI. That's basically what we're doing. We're talking about making machine learning models. We're just talking about really AI here, right? But we need to be feeding those models actual experimental data. And so the better we can make these test sets, the more data we can feed, the better these algorithms are going to get, the more control we'll have, and less thinking and experimental trial and error we'll have to do.
And I think that that's really the power of AI is not the algorithms per se, but the underlying data, making sure we have data that is widely distributed in the sequence function space for searching on, and then address it from codon bias, codon sequence, initiation transcription, initiation translation. And I think what we've done on the mammalian side is we're using transposomes now to be able to integrate lots of copies. And I think there's going to be a number of areas where, at the end of the day, there are only so many levers to pull. There's replication, transcription, translation, and degradation. That's kind of Wilson Crick sorted that out, what is it, 70 years ago or so. But there's still a lot to learn. So I think we are closing into our full hour, and do a handover back to Aisha and see where. And I really appreciate, Dom, you taking the time. This is awesome to be on the same webinar as the king of expression. So that's really very cool. Really appreciate that.
Yeah, thanks. It was great.
All right, thank you so much to our panelists for that absolutely wonderful presentation. We hope our audience gleaned a lot of insights and learnings from this presentation, and thank you once again to our expert panelists. We have now reached the end of the webinar. If we couldn't attend to your questions during the Q&A session, there were a lot of very interesting questions that came in. The team at ATUM may follow up with you, or if you have any further questions, you may direct them to the email address displayed on your screen. Thank you everyone for participating in today's webinar. You will be receiving a follow-up email from Xtalks with access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve our webinars. In addition, I'm just going to go ahead and share a link in the chat box. Oh, let me just share that link right now in the chat box. There we go. And you can view the recording of today's event at that link, and you can also share it with your colleagues so that when they register for the event there, they can also view the recording. So I do encourage you to do that. For those watching via LinkedIn Live events, we encourage you to visit xtalks.com to register for this event and gain access to features such as viewing the recording and more. Now, once again, please join us in thanking our speakers for their time and insights today, Dr. Esposito, Dr. Gustafsson, and Dr. Welch. We hope you enjoyed the webinar once again. And on behalf of the team here at Xtalks, thank you very much for attending, and we hope to see you next time. Have a great day, everyone, and thank you all once again.